Right. Let's move on then, and I'd like to pass on the floor to Pavel Shkarlat, DSM Nutritional Products, Moscow, and he will tell us about the molecular genetic analysis methods to evaluate the poultry store symbiotic impact on the microbiota on broilers. Thank you so much. Dmitry Spiridonov, who delivered his presentation on Amorimidase, which is a commercial product offered by DSM, which is an undoubted leader on feed and animal health. Well, my presentation will supplement his presentation. Just a few words about the company. We are experts on the various feed premixes, and since premixes are closely linked with the health status, we also focus on that. We pursue three work streams: vitamins and premixes, that's the first one, and DSM. I used to call Hoffmann-La Roche, and it was a pioneer who chemically synthesized vitamins, and we've been working in this domain for over 70 years. Premixes that we produce in Russia take into account all the national features of our poultry farms. We deliver high-quality products that solve economic problems facing poultry and the pig farms in Russia, as well as carotenoids. Well, we talked about the antioxidants and improving the fertility. So carotenoids are well known to vets and flock managers. The second group of products that we actively sell in Russia are food supplements and enzymes. Well, as far as enzymes are concerned, we already mentioned Meramidase, that is part of the enzyme products that we offer. These are high quality products, thermally stable, and they help farms to solve economic problems through optimizing diets, since they contain contemporary enzyme combinations. Mycotoxin deactivation products is the next category. Mycotoxin absorption is not always effective. And out there in the market, we see more and more products that are linked with the enzyme deactivation of certain types of mycotoxins. And we believe that the mycotoxicosis control will focus on that in the future. Eubiotics is something that my presentation is devoted to, and the so-called special nutrients are the products that ensure better digestibility. For instance, the HYD, which is a derivative of vitamin D, manufactured by our company and used by many farms. The third work stream is lab solutions, and we identify mycotoxins with the help of the tandem mass spectrometry in our lab. Up to 32 mycotoxins can be identified within a single run at our lab. In addition to that, we analyze raw materials in the infrared spectrum, and we also do we also do NGS sequencing to analyze gut microflora. Thank you for your attention. So anyway, as far as the experiment is concerned, I met one of the owners of a very large poultry farm that uses one of our products to optimize their performance indicators, and he told me, fine, I see that the KPIs are going up, the survival rates are going up, the FCR goes down, but based on evidence-based veterinary medicine, we were talking about a probiotic, by the way. Can you prove that it impacts the gut flora and changes its composition? We initially planned to carry out this experiment in abroad, but then we decided to run it in Russia. We did it jointly with the poultry industry department of the Moscow Veterinary Academy. To be able to talk about the changes in the gastrointestinal tract, I just wanted to first say a few words about the microbiota and what it influences. Well, lots of vets and practitioners are following us online, so I'm gonna give you more details on that. Microbiota is a microbe organ that has direct and indirect impact on metabolism, behavioral functions, signal mechanisms of a body. We all know the role of the composition of the gut flora on the immunity, because 70% of the cells, immunocompetent cells, are in the gastrointestinal tract, and naturally the microbiota composition has a direct impact on the immunocompetent tissues and organs. It also has an impact on synthesis and production of microbial substances and compounds, protect the epithelium, and penetration of pathogens into the bloodstream, the forming of colonies. The impact on the metabolism through diet, since birds have no endogenous enzymes that are necessary for digesting non-starch polysaccharides. Microbiota plays a huge role in digesting such components. Vitamin B biosynthesis, vitamin A biosynthesis, electrolyte balance. However, oftentimes we tend to forget that the gut microflora also takes part in dopamine synthesis and serotonin synthesis, that in their turn have a calming effect, therefore affecting the central nervous system. Ultimately, you can establish the link between the gut microflora and the behavioral reactions of a body. And this was mentioned time and again. Susceptibility to stress in the flock. So what is microflora, how do we study it, and so on and so forth. Different publications mention that microorganisms could account for 50% of the dry feces. And according to various experts in different parts of gut, you might find 300 to 10 to 3 to 1000 different microorganisms, for instance, in the cecum. 30 to 40 types account for 90% of the microorganism population. What microorganisms do we study primarily? What are their functions and how do they interact? While in the past, we depended much on the golden standards in microbiology. Now we can use microbiology such as DNA sequencing and various statistical models and software suites that help us identify such microorganisms. what techniques did we use in this examination? Real-time PCR, that's first and foremost NGS sequencing. In TRFLP, we identify the composition, the number, the quantitative and qualitative composition. To what purposes did we apply these techniques during the examination? Well, first and foremost, to identify the status of the gut flora and identify diseases in even those at the subclinical stage, develop the focused regulation techniques to regulate gut microflora, and last but not least, identify the efficacy of such products if they are administered. For instance, if we're talking about a probiotic, we should see a definite effect on the gut microflora. The objective of the study was to identify the impact of symbiotic, which goes by the commercial name of poultry star, on changing the gut flora in broilers. The composition includes Enterococcus faecium, Bifidobacterium animalis, and Lactobacillus salivarius. And as was mentioned by Vasily, the proper lactobacillus is the one that when it goes into the gut, persists there, proliferates there, and occupies a niche and stays there for a prolonged period of time because it's a symbiont of an intestine. And we used fruit oligosaccharide as the prebiotic. There were two groups, the experiment group and the control group. The only difference was the probiotic administered. We slaughtered the broilers, sampled the cecum content, and we used the sequencing, NGS sequencing, and then analyzed the results with the help of the statistical software suite. The first thing I wanted to draw your attention to is that the overall count stayed the same in the control and the experiment group, despite the administration of the drug. The number of microorganisms remained the same, and that just goes to show you that the count was not affected, while the composition was. This slide shows the outcomes of the research done with the help of the NGS sequencing. We split here the results into two groups, control and the experimental group, and they're split into opportunistic pathogenic organisms and non-pathogenic organisms. I will only dwell on the statistically significant organisms shown here. Spirochetes. These are pathogenic or opportunistically pathogenic organisms. The share of Spirochetes went down in the experimental group as compared to the control group. Lactobacillus antagonists of Spirochetes, in the experimental group, their number increased by 4.3%. As for bacteroidetes and firmicutes, they take an active part in digestibility. Like I said, birds lack enzymes for digesting starch, polysaccharides, and cellulose. Both of these bacteria produce such enzyme. Therefore, their greater number helps digestibility. In both groups, we saw that the number of these microorganisms This is real data. So as for actinobacteria, as for bifidobacterialis and actinobacterias, the share was not that significant, but they still play a huge role because they produce antibacterial substances. And what we saw was the greater number of actinobacterias. The increase was 0.15%. To sum up, symbionts, symbionts count and opportunistic pathogenic microorganisms. The figures are shown in the slide, and in the experimental group, we saw an increase in the symbionts, while in the control group we saw a increase of pathogenic or opportunistically pathogenic microorganisms. We talked about the stress factors that impact the growth rates. I'm not going to go into details here, but most of them have an impact on the composition of the gut flora, either directly or indirectly. And if you want to help your flock cope with certain stresses caused by unfavorable living conditions, and you want to unleash the potential of the broilers to the maximum extent possible, and symbiotics or drugs based on microorganisms have proven their efficacy. In conclusion, I wanted to point out that new diagnosis techniques are available. You can study the gut flora with the help of these new methods. And the new molecular biology techniques should be actively used, at least I believe that, at poultry farms and vet labs. And when you choose a drug that you want to use, please be guided by evidence-based veterinary medicine. Thank you for your attention, and I'll be happy to take questions either now or later on offline, or you can forward them to me by email. Thank you so much, Pavel. Ladies and gentlemen, any questions from the floor? Well, you can post your questions in the chat box, and you will stay for gallery dinner, right?